The today's topic is electrophoresis. That is very much similar to as that of electrolysis. Electrophoresis that is used to diagnose the different type of amino acid, especially when the DNA fingerprint or the DNA different DNA test, or whenever you are going to hydrolyze the protein, and there is the formation of amino acid in different samples or different amino acid in one sample. So I'm going to talk about electrophoresis. Basically, electrophoresis is a technique, as I told you, to that is used in the laboratory to separate the different amino acid based on their size and charge. I mean, the most important factor in this case is this one, that different amino acid can be distinguished on behalf of their sizes, on behalf of their charge. But first of all, these amino acids are separated, or sometimes the protein is hydrolyzed into acidic or alkaline medium. And here is a general apparatus for the further electrophoresis. Electrophoresis once again mean electro mean electricity, and phoresis mean to, to carry out them, to transport them. Electrophoresis, the apparatus, simple electrophoresis apparatus is over here, in which, for example, this is the battery, DC battery source, negative terminal, and this is the positive terminal. This is one beaker or one tub, you can say, that is having electrolyte. Same electrolyte is present on the other beaker, but this rod is dipped, this metallic wire is dipped into this one, into one beaker, and other one beaker is acting the other electrode. So this beaker is acting as negative terminal or negative sphere, negative portion. This beaker is going to act as the positive terminal over here. Here is the glass slide or glass plate that is basically connected or enveloped with the moist what paper, that is roughly, you can say, the filter paper, and a mixture of different amino acid is situated over here at this spot. So whenever we are going to start this uh, procedure that is called electrophoresis, then a sufficient or concentrated spot is basically placed or based on the middle of that paper, that is basically present between, that is sandwiched between two glass slides. When the current is switched on, then some of the amino acid molecules or ions, deuteron, move towards the negative if they are having positive terminal, positive charges. Some of the amino acid molecule or ions will move towards this tank, this beaker, this positive terminal. Those are having negative, overall negative charge on it. So there are the main components of electrophoresis, and you can see it over here. And as I told you, the spot is placed at the middle of this slide, and basically between these two slides there is a filter paper that is soaked with the same liquid, same electrolyte that is present in both beakers. Now, there are different things, different factors. Those are going to govern the speed of and the direction of this amino acid or these amino acid. Those are present in the mixture. It is also similar to as that of the paper chromatography. As you have seen that on the paper chromatography, there is a traveling of different spots on the paper uh, because of or due to the solubility or the difference of the solubility. Here the movement is because of the dragging force of current, electric current, DC current is dragging them. Some are going to be dragged towards the negative terminal, and other ones are going to be dragged towards positive terminal. So that is, here is the journal description of this whole procedure. Electric field is applied, number one, with the force of attraction between the electrodes. Oppositely charged component equal to the is equal to the fraction of the force. The voltage of the battery is enough stronger that is going to dominate the friction of uh, the paper that is provided by the paper, by the filter paper, so these spots can travel easily to minimize the friction on the paper, and they can move towards either positive or negative terminal. The next thing is there is a gel medium resisting the motion of that particular species. Relative rates at which component mean amino acid, basically components mean amino acids or amino acids, what you can call it, move, and therefore extent to which they separate are influenced by the strength of the field. Once again, as I told you, the basic main driving force that is going to separate them out is the voltage given by this direct source, DC source. So that is going to give enough force to travel, to drag them out. That is more than that of the friction that is created by the movement, or that is given by the filter paper to the spots or to the amino acid. Each species responds to this factor according to which is called electrophoric mobility. There is a special term that is said to be electrophoric mobility. This is a constant for the each species. Now, there are the different factors. Those are going to affect on the mobility of amino acid. Number one, voltage. Once again, voltage, as I told you initially, basically the voltage of the battery, if it is higher, that is directly proportional to the its moving gradient, its moving speed across the gel or across the paper, whatsoever is the medium. So voltage is directly proportional to the rate of speed of movement of amino acid. Next thing is the size. Smaller the size of the molecule migrate quickly than that of the large molecule. So small size amino acid travel more faster as compared to the large size. Third one is the shape. Shape mean it may be irregular shape or it may be the regular shape. So if a molecule with a lot of side chain experience more friction resistance by the paper, by the gel. So, an irregular shape or the molecule that is having with more chains, more side chains, it will travel slowly as compared to the molecule that is small in size and that is regular in shape, that is having less number of side chain, it will move quickly. Fourth, and the next important factor is this one, basically buffer of the pH. I mean, as we are using the electrolyte, basically these electrolytes are buffer cells. I mean, they are acting as a buffer solution. These two solutions are, in fact, indeed, they are buffer solution. Those are having the same pH throughout the procedure. So the pH of this solution also matter. If the pH is alkaline, then it might be possible, then the maximum population of the amino acid is going to have negative charge on it and they will be moved toward the positive terminal. If we are going to change the pH towards acidity, then it might be possible that the most of the amino acid or all of the amino acid are going to have positive charge and they are moving towards negative terminal. So the direction or the mobility, you can say, basically direction, it is also going to depend on the buffer's pH. As I told you in my previous lectures, that the deuteriene like amino acid, they are going to be establish themselves in form of deuteriene. They can be either positively or negatively ionized, charged, because they contain both acidic and basic group. The extent and the direction, both ionization, depend on the pH of the buffer. Basically, as far as my opinion is concerned, basically pH changes the direction of the amino acid. The fifth one is the temperature. A rise in temperature can speed up the electrophoresis, but can also denature the protein. So if you're going to speed up the reaction, I mean, if you're going to increase the temperature, reaction will be speed up, their movement will be sped up, but that protein that is going to be examined over here, it may be denatured. So as far as the general experience is concerned, temperature must be optimum. It should be normal at the normal level. Then direction, the next further main thing is this one. Direction of the movement of amino acid. either they will move towards negative terminal, or will they go towards positive terminal. It is going to depend upon the basically, number one factor is pH, as I told you, in the pH of the amino acid and the resultantly charge on the amino acid. So actually that is one and the same thing. If you're going to change the pH will change the overall charge on the amino acid. I mean, change in pH mean if you are going to change from acid to alkaline medium into alkaline pH, then definitely overall charge on that amino acid group or these amino acid groups will be changed. So that is only one factor in my opinion, but in different books it's written as a two factors. So one is the pH and other one is the overall charge on these pH. So these are the basic theme, main uh, points postulate to remember about the electrophoresis of amino acid, and this whole separation, this procedure is called electrophoresis. For example, here is a work example. I'm going to solve it. A mixture of amino acids may be separated using this one, I mean this technique, electrophoresis. A typical practical setup is shown in the diagram. That is the similar one that I show you on my previous page. Now what is the question? Basically, here is the question. When a power supply is switched on, some amino acid may not move but remain stationary. Suggest an explanation for this observation. This for 2 marks. So, what is the reason behind it? Some amino acid, they are not moving towards either towards negative terminal or towards positive terminal. They are not showing any sort of movement. What is the reason behind it? Can you suggest the reason? Explain the observation.
Neutral or strong bonds.
Correct. Number one, these amino acids are having basically neutral. They are isoelectric, I mean, isoelectric, having same charges. I mean, overall, they are neutral, isoelectric, having same charges on them. And so they are having equal force of attraction, equal driving force from the both terminal. So that's why, where is my cursor? So equal force EMF will experience on them from both sides. Experience on them from both sides. Both sides are both terminals. So that is the appropriate answer. Amino acid glycine has the formula this one. Identify the species formed on the filter paper if the glycine moved towards the left, toward the positive, and on the filter paper. Here is using the filter paper basically. So if it is moving towards the positive side, then it must be having one negative charge on it. So this species will be like that: NH2, NH2, CH2, C double bond O, O negative ion. In this molecule is no more deuterine. It is having only one negative charge on it, right? One more thing, there is one another possible answer of the above thing, that as I write it over here, they are at isoelectric. Uh, there must be another factor that is the same one and same thing. I mean the pH of the buffer, pH of buffer is at isoelectric point. That is one and same thing. Basically, why they are going to be at isoelectric? Because they are having, I mean, their pH is going to make them isoelectric. Now they are going to have same positive and same negative charges. These are basically because of the pH of the buffer. So you can write this sentence as well. Now, see, following result was obtained from another electrophoresis. Following, sorry, this not over here. What can be deduced about the relative size, charges, and I think it's on the next page. Yes. Once again, here are the results. Initially, the spot was present over here. So it's B, A, C. Two spots have been moved towards the positive terminal. One spot moved towards the negative terminal. Mixture placed over here, as I told you. And they are asking to write the sizes and relative charges. Charges are very easy. A is having negative charge because it is moving towards the positive terminal. B is also having negative charge as it is moving towards positive terminal. C is having positive charge because it is moving towards negative terminal. Now come to the next one. Which one is the relative size? Small,
बी
large, larger, or medium, you can say.
सिर्फ बी लार्जेस्ट है, उसके बाद सी मीडियम है और ए स्मॉल है.
Good boy. A is the small in size, and B is the large in size, and C is medium in size. Because this distance from the center will let you know which one is showing the higher speed. Higher speed mean smaller the mass. Speed and mass are inversely proportional to each other, right? Now I'm going to solve electrophoresis question, and here is a data booklet of electrophoresis. Basically, these are the side chain. For alanine, this is a side chain. For aspartic acid, ASP, this is a side chain. For cysteine, this is side chain, and so on. So, what is the question? Question number 1. Use a data booklet to draw the structure of tripeptide ALA serine glycine showing its peptide bond in full. So first of all, I'm going to write alanine because on the left hand side there's alanine, then serine, then glycine. As I told you, the basic structure of all amino acid is this one, C double bond O, OH, and here is the carbon, here is nitrogen and hydrogen, and here is the hydrogen, and here is the R group, side chain group. So in case of alanine, the side chain is basically CH3. Methyl group is on the side chain, so I'm putting CH3 over here. Then there is serine, again H, N, H, carbon, hydrogen, C double bond O, OH, and in case of serine, the side chain is CH2OH group, serine. Alcoholic group is over here. So CH2OH. What is the glycine? This is the smallest one and the smallest amino acid. Here is the hydrogen, here is again hydrogen, and C double bond OH. Once again, let me check it over here. This is the glycine that is having just hydrogen as a side chain structure. So now how to make the peptide linkage? By removing the OH group from acidic part, H from NH group, and there is the formation of peptide bond. The second bond is going to be formed over here by the similar way. And finally, I am going to erase these bonds and erase these molecules and making the proper bond between C and N. Same is the case over here. There will be a proper bond between C and N. So CN bond. Here is again CN bond. So there are the two, basically these peptide bond. Overall 3 amino acids are there. That's why it is called tripeptide. Tripeptide mean 3 amino acids are going to meet to make a linkage to make a peptide bond. The next question is calculate relative molecular mass MR of ALA, serine, glycine. So I am going to count their masses. How many NH over here? 2 hydrogen, 2 into 1 plus 1 nitrogen, 14. There are 1, 2, 3 carbon, 12 into 3 plus 3 plus 1, 4, and 4, 5, 6. 6 overall, total 6 hydrogens are there in this unit. Here, let me write once again. I'm going to write hydrogen first, that is having 1 AR, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, and 15. So 15 hydrogens are there. How many nitrogens are there? 1, 2, 3 nitrogens are there. So 3 into 14 for nitrogen. How many carbons are there? 2 carbon, 3 carbon, 3 carbons. 3 plus 3, 6 plus 2, 8 carbons are there. 8 into 12. Plus how many oxygens are there? 1, 2, 3, 4, and 5. 5 oxygens are there. So that is this sum is equal to the MR of this whole tripeptide. That is equal to 233 if I am not wrong. Electrophoresis can be used, part B. Electrophoresis can be used to separate the mixture of amino acid and peptides. A mixture of tripeptide, ALA, serine, glycine, and its 3 constituent amino acid were subjected to electrophoresis in a buffer at the pH 11. Draw the structure of serine at pH 11. So, this is a serine, this one structure, the middle one, this one. And what will be its structure at pH 11? pH 11 means it's an alkaline. So in case of alkaline medium, first of all, I'm going to write the normal amino acid serine, that is CH, CH2OH, C double bond OH, NH2, NH2. So in this case, generally, if I'm going to add alkali, pH is equal to 11 mean alkali. So this is the acidic part, the only acidic part in this molecule that will react, and it will lose H positive ion. So remaining over here, H positive ion will be lost and O negative ion will be formed. So finally, this is the structure of serine, that is, and now it has become an ion in pH 11. Next part, at the end of the experiment, following results were seen. There are different alphabets: F, H, I, S, R, Q, B, G. Identify the component labeled F to I in above diagram. What is F? F is the basically battery source, as you have seen in my lecture. It's a DC source, direct current source. Direct current source. G is the either positive or negative terminal. The next thing is H. Again, it may be one of the positive or negative terminal. I is the filter paper. That paper, that is a base for the mixture to be examined. So the filter paper suggests the identities. First of all, what is G and H? You can see that the maximum spots or all spots are moving towards P. If all spots are moving towards P and one of the spot we have diagnosed at pH is equal to 11, that was anion. Serine was anion. So obviously serine is one of them. So this terminal G will be the positive terminal. Positive terminal, we can call it anode. So obviously the H will be negative terminal. It will be negative terminal, that is said to be cathode in this case. And I is the filter paper or any, medium, you can say base medium, sporting medium for the sport. I'm writing filter paper. Now, the second part is identities of the species responsible for the spot P and S. P is the highest moving spot over here, that is moving very speedily, and S is the slowest spot moving over here. So basically, which one thing will allow them to move faster or slower? That is generally charge to the mass ratio. Charge to mass ratio. So basically, charge over mass ratio is the main factor, as the main clue, main key to find the speed of any amino acid on the filter paper that is traveling towards either positive or negative terminal. As you have seen that we are having DC source, and this is the positive terminal, and all are moving toward the positive terminal on this filter paper that is soaked in buffer solution. Filter paper soaked in buffer solution. So charge to mass ratio, all these species are having one negative charge, as we have seen in case of serine. And similarly, if I'm going back on this picture, you can see that if I'm going to put this tripeptide into an alkaline solution when pH is equal to 11, then there is again only one acidic group lying over here that will react with the alkali, and here is the presence of one negative charge. There is no any other group that is going to react with alkali. So this whole huge molecule is also having just one negative charge. So all are having one negative charge, or all will be having one negative charge. Alanine, serine, glycine, all will be having one negative charge individually. And in this tripeptide, this is also having one negative charge. So charge to mass ratio, basically they are having one negative charge, all of them, and here is the difference of the mass. So higher the mass, slower the speed of traveling. So this S is having, it means that this is having, as it is having slowest speed, so it is having highest mass. Mass and speed are inversely proportional to each other. So this S will be the tripeptide that is having the highest speed, that is tripeptide. That was ala, dat serine, dat glycine. So what is the P? That is having the highest speed, so its mass will be the lowest one. So which one is having the lowest mass? That is having lowest side chain, or the minimum side chain, and that is having this one, glycine. This one negative ion, this one negative ion of the glycine. When we will make the negative ion of the glycine, that is an HCH, both are hydrogen. I'm going to write CH2O, and here will be negative because of the pH of 11. So this is the negative ion that is in fact P, that is having the lowest mass. Glycine or this one negative ion, both are the correct option. So NH2 CH2, C double bond O negative ion is the P. Explain your answer. As I told you, basically they all are having same charges, and charges are same on all of them. Charges are same. Number two, they are different in masses. Different masses. So as I told you, the speed is directly proportional to charge over mass answer. So that's all about this question. Thank you. Thank you very much. Please subscribe my channel, hit the bell icon and share it with your friends.